Behavioral Neuroscience, Chapters 9 and 10, Segment 2. Central deafness is hearing loss caused by brain lesions, such as a stroke, and it's seldom a simple loss of auditory sensitivity. Word deafness is a disorder in which people show normal speech and hearing for simple sounds but cannot recognize spoken words. This may be due to an abnormally slow analysis of auditory inputs. Cortical deafness is patients have a difficulty recognizing both verbal and nonverbal auditory stimuli. It's a rare syndrome because it requires bilateral damage to the auditory cortex. Treatments for deafness focus on replacing missing stimulation. Electrical hearing aids are miniaturized audio amplifiers. A microphone picks up environmental sounds, amplifies them, and plays the amplified sounds directly into the ear canal. They can't help as well for sensory neurohearing loss, where the hair cells and other neural components of the inner ear are damaged or missing. Cochlear implants are an electromechanical device that detects sound and selectively stimulates nerves in different regions of the cochlea via surgically implanted electrodes. This provides only a limited range of frequency and loudness. They don't restore normal hearing, but it provides stimulation to the brain that can be interrupted as sound. The receptor mechanisms for the vestibular system are in the inner ear. The components of the vestibular system are a part of the inner ear attached to the adjacent cochlea with a hollow in the temporal bone. There are 3 semicircular canals, which are fluid-filled tubes oriented at right angles to each other. Nodding up and down is pitch, shaking side to side, and tilting your head left to right. The 3 canals are connected at their ends with a sac-like structure called the auricle. Below the auricle is another small, fluid-filled sac called the saccule. Receptors here are groups of hair cells whose stereocilia bend, leading to the excitation of nerve fibers. Each canal terminates near the auricle in an enlarged region called the ampulla that contains the hair cells that signal movement in the corresponding planes. The stimuli of the hair cells are embedded in a gelatinous mass called the cupula. On the cupula, the small bony crystals are called the otoliths, and they increase the sensitivity of the receptors to movement. The vestibular system evolved from the lateral line system, which is a sensatory system found in many kinds of fishes and some amphibians. Nerve fibers from the vestibular portion of the vesicular nuclear nerve, which is V3, so 8, synapsize in the brainstem. Nerve fibers from the vestibular nerve enter lower levels of the brainstem and synapse in the vestibular nuclei. Some fibers bypass and go directly to the cerebellum, one important function in the precise control of eye movements. The VOR, the vestibulo-ocular reflex, the brainstem mechanism that maintains the gaze on the visual object despite movements of the head. These functions are accurately used when eyes are closed, showing vestibular inputs rather than visual ones. Some forms of vestibular excitation produce more motion sickness. Motion sickness is caused by movements of the body that we cannot control. Sensatory conflict theory is discrepancies between the vestibular information and visual information, which stimulate food poisoning and trigger nausea. A big controversy is with airplane pilots. Linear acceleration and an upward head tilt produce the same stimulation of the otolith organs. In conditions of very low visibility, an acceleration of a plane may be misinterpreted as a climb. The false climb illusion leads pilots to force their planes into a dive because they believe that they are climbing. Pilots making the landing in low visibility conditions learn to rely on a variety of instruments rather than on their vestibular system for this reason. So taste excites specialized receptors on the tongue. Papillae are the little bumps on the tongue, and they are lumps of tissue that serve to increase the area of the tongue. There are 3 types. The circumvallate papillae and the foliate papillae contain many taste buds on its sides. The fungiform papillae contains only about 6 taste buds each, and they look like little button mushrooms. Each papilla holds 1 or more taste buds, and each taste bud consists of a cluster of 50 to 150 taste receptor cells. At the surface end of the taste bud is an opening called the taste pore. The taste cell extends fine cilia into the taste pore, which come into contact with tastants, all of which are substances that can be tasted. Each taste cell is sensitive to one of the 5 basic tastes and have a lifespan of only 10 to 14 days. They contain immature taste cells, which eventually become the new taste cells. Different cellular processes transduce the different basic tastes. Salty. Sodium ions from salty foods enter taste cells via sodium channels in the cell membrane, causing a depolarization of the taste cell and release of the neurotransmitter that stimulates the different neurons they relay the information in the brain. The second salt receptor, which is a variant of TRPV1, may provide additional sensitivity to salt and cantons from other salt in foods such as potassium and calcium. Sour. Acids in food taste sour. Acids release hydrogen ions. The acid-sensing ion channels resemble iotropic receptors and have been proposed for sour detection. One type of potassium channel that is blocked by hydrogen, preventing the release of potassium ions from taste cells and causing depolarization and neurotransmitter release. Sour-specific taste cells need one type of ion channel, PKD2L1, to function, which is also detected in carbon drinks. Sweet. It appears to stimulate specialized G-protein coupled receptor molecules on membranes of the taste cells, resulting in the intercellular activation of second messengers. These receptors belong to the T1R and T2R families. When 2 members of the T1R family, T1R2 and T1R3, combine, they make the receptor that detects sweet. The different sweet tastes appear to interact with different recognition sites within the T1R2 plus T1R3 receptor complex. Bitter. Members of the T2R family of G-protein coupled receptors appear to be bitter receptors. The T2R family has 30 members. Each bitter sensing taste cell produces most or all of the different types of T2R receptors. They are all maximally sensitive to any of the wide variety of bitter substances, but are not very good at encoding qualitative distinctions between them. Behavioral and electrophysical data indicate that at least certain broad classes of subjectively bitter substances are discriminable. Umami. 2 types of receptors are specialized to respond to this taste. One is a variant of the metropic glutamate receptor. It is expressed in certain taste buds and most likely responds to the amino acid glutamate, which is found in high concentration in meats, cheese, kombu, and other savory flavors. Second is the heterodimer of the T1R3 receptor. Taste information is transmitted to several parts of the brain. The gustatory system, which is the taste system, extends from the taste receptor cells through brainstem nuclei into the thalamus and to the cerebral cortex. Each taste cell transmits info to several different fibers, and each fiber receives information from several different taste cells. The 8 different fibers rub along 3 cranial nerves: the facial, the glossopharyngeal, and the vagus nerves. The fibers in each nerve run to the brainstem, where they synapse with second-order gustatory neurons that project to the ventral posterior medial nucleus of the thalamus. After another synapse, the third-order gustatory fibers extend to the cortical taste area located in the insula, just anterior to the central sacculus. Some gustation researchers argue that the brain exploitates taste info from pattern coding, the related activity across ensembles of axons from different taste receptor cells. This provides great capacity for distinguishing subtle differences between stimuli, and produces a rich parallel set of inputs upon which the brain can act to tune the stimulus it receives. Other researchers believe that taste sensation involves a simple system of labeled lines, in which there are 5 distinct types of axon lines, corresponding to the 5 classes of taste receptor cells. 1 line for each taste. Selective and activating cells that express receptors for just one of the 5 tastes tends to eradicate sensitivity to that one taste, while leaving the other 4 tastes unaffected. This can establish whether it's a pattern line or a labeled line. Insomnia is odor blindness. The sense of smell starts with the receptor neurons in the nose. The olfactory epithelium is a sheet of cells that lines the dorsal portion of the nasal cavities and adjacent regions, including the septum. There are 3 types of cells: supporting cells, basal cells, and olfactory receptor cells. Each olfactory receptor cell has long, slender, atypical dendrites that extend to the outermost layer of the epithelium, the mucosal surface. The cilia emerge from the dendritic knob and extend along the mucosal surface. At the opposite end of the bipolar olfactory receptor cell, a fine, unmyelated axon runs to the olfactory bulb. Odorants excite specialized receptor molecules on the olfactory receptor cells. The direction of the airflow in the nose is determined by the complex curved surfaces called turbinates that form in the nasal cavity. Airborne molecules initially encounter the fluids in the mucosal layer, which contain binding proteins that transport odorants to receptor surfaces. The odor stimulus then interacts with the receptor proteins located on the surface of the olfactory cilia and the dendritic knob of the receptor cells. Receptor proteins are members of the G protein-linked receptors. Interactions of odorants with their receptors trigger the synthesis of second messengers, such as cyclic AMP. Cyclic AMP opens caution channels, which leads to the generation of action potentials. Olfactory axons connect with the olfactory bulb, which sends its output to several different brain regions. The olfactory bulb is an anterior projection of the brain that terminates in the upper nasal passages and through small openings in the skull. It is organized into many roughly spherical neural circuits called glomeruli, within which the axon terminals of the olfactory neurons synapsize on the dendrites of the specialized mitochondrial cells of the olfactory bulb. Olfactory receptors are found in the axon terminals of olfactory neurons as well as in their dendrites. Output from the olfactory bulb consists of the axons from mitral cells, which extend to various brain regions, which include the prepiforum, the orthognathic cortex, the amygdala, and the hypothalamus. Functional MRIs that are studied suggest that the human prepiforum cortex is activated during sniffing, whether or not the odor is present, because the airflow provides somosensory stimulation. Many vertebrates possess a veterinasal system. Pheromones are odor signals or trails that many animals release. The ventronasal system detects pheromones. The receptors for the system are found in the ventronasal organ, the VNO, of epithelial cells near the olfactory epithelium. It sends their information to the accessory olfactory bulb, which projects to the medial amygdala, which projects to the thalamus. Humans have a structure that resembles the VNO and show behavior evidence of some pheromone-like capabilities. Genome testing indicates that genes that encode V1R and V2R are non-functional, as is the gene that encodes the TRP2 ion channel. TAARS, or trace amine-associated receptors, are a family of probable pheromone receptors produced by neurons in the main olfactory epithelium. This is a new class of olfactory receptors which is found in rodents. The cutting edge, more than a matter of taste. The taste, like chemosensation, is wide throughout the body, since finding of taste-sensing cells, such as the taste-associated G protein gustductin and the T1R and T2R based taste receptors. Cells in the stomach and the intestine express gudductin and T1R3 sweet receptors to help organize appropriate digestive responses. Cells in the stomach and the colon express T2R bitter receptors that may alter the transit time of foods or cause diarrhea. By altering the release of appetite-related hormone ghrelin and inhibiting stomach activity, bitter taste cells of the gut may productively adjust appetite and slow the absorption of ingested material. Taste-transducing cells have been found in the airways, primarily T2R bitter receptors, which appear to regulate productive reflexes like sneezing and coughing. The role of bitter taste receptors in the testes remains mysterious, but elsewhere they sense the toxins.